So what we were talking about at the end is a very generalized idea for how disease resistance works in plants. So first off, if you didn't know this, plants have disease resistance properties. They're actually pretty good and pretty robust. Okay? And you can think about them as being like the innate immunity in humans. Okay? So they sense something and they respond to it. And one of the ways, the biggest, easiest to see response is basically localized cell death to try and control wherever the pathogen has gotten in. So you have a leaf, it gets infected, and that infection can spread. But if you just kill off the cells right where that infection started, then your infection's over. Okay? So what plant pathologists have studied for a really long time is how does that happen? And breeders are obviously very concerned with that as well, because they want to pick that or engineer for that in their crop. Alright? So that's the very broad background. So he was talking about Fusarium and about resistance within cotton and Fusarium. So you have your plant and you have your pathogen, and when they interact, you want the plant to shut down the pathogen, right? Because you want the cotton to be able to grow and not die. Okay? So that's what he's studying. Various races of pathogens, you can kind of think of just being, they're really similar to one another, so it's still the same species, but sometimes they infect slightly different types of plants. So it can be something as simple as the two types of cotton he was talking about, the Pima and the upland. They're a little bit genetically different as well. So one race might grow really well on upland, and another race might grow really well on Pima. Okay? And one race might not grow at all on Pima, because it's just different enough to have a resistance gene, to have resistance to that race, but not to another one. So we're talking about really small genetic differences most of the time. And we're also talking about, so this is where the discussion went at the end, the idea of a gene for gene interaction, which means you have gene A in your plant, and it provides resistance to pathogen 1. Pathogen 1 comes on that plant, gene A responds, pathogen can't grow. Okay? So that's a gene for gene response. This is really similar to a lot of what has happened in terms of human health. If you think about the fact that like 15 years ago, people said when we sequence the genome, we'll cure everyone of everything. Turns out there's a lot of factors involved in curing human diseases. Very similar in plants, right? Sometimes you get lucky and you get this gene for gene thing, right? And that's the same as having like one gene that's causing breast cancer. Okay, but mostly everything involves multiple different loci. And that's that QTL term, which you guys have probably come across in genetics class or some other classes you may have taken, right? But that's quantitative trait loci. Basically what it means is when you have a lot of genes doing a little bit of work, you can end up with a big response. So you can't isolate your response back to any single gene. You need a mess of them to all work together. Okay? So that, as you can imagine, and as you may well know from the human world, makes it really difficult to give a direct diagnosis of this is what's wrong, here's the gene we need to fix it, because you have maybe 20 genes that are involved, all giving a little bit here or there, right? So you don't get super lucky and get like the one like slam dunk gene. You're looking at a lot of different things. Okay, so that was the PCR he showed us. He had a PCR that had, you know, a bunch of different individual plants and 2 markers, one that was on the top and one that was on the bottom. And one of his genes that he's looking at, or it might not be a gene because he doesn't know exactly where all the genes are, but one of his markers, so indicating there's a gene there that's doing something, has like a 70%, you heard him say that, association. So that means about 70% of the time, when you have, let's say, the bottom allele, because I don't remember what it was, you're resistant. And if you have the top allele, you're not resistant. Sometimes you can get the bottom allele and still not be resistant. All right? So it's an association, and that's what makes all of this super messy, okay? And really hard to try and parse out exactly what's going on. So that's why he's going to use, so MySeq is this machine from Invitrogen that basically will give you good sequencing data. So you can sequence 100 different individual plants, look at exactly where they have different bases, and then you use the supercomputers he was talking about to try and make sense of all that. You map all of that on top of each other, you look for patterns. When you find a pattern that you think fits, you move on and try and find another pattern, basically, okay? And in QTL situations, you might be looking for, 50 or 100 different spaces along your genome where this is happening. Did any of that help? Okay. I will look at my notes. Do you guys have